My name is Josh Bishop. I am the Global Chemistry Account Manager for Chemistry, Pharmacology, and High Throughput Screening, IT, and Informatics at Merck Research Labs, IT. So what does that mean? It's a very glorified title, but it means that I take care of chemists from discovery medicinal chemistry all the way out to early development in my process and chemical engineering labs. So a lot of end users to look after and a lot of technology to look after. One of those groups that I do support is called the Chemistry Modalities Organization. And so that's a spin-off from our Discovery Chemistry organization. And they started up probably in about August of last year. So right around the time that I joined up with MRL as well. And they are looking at non-traditional drug targets and drug tool compounds. So they're looking at biopolymers, peptides, oligonucleotides, antibody-drug conjugates, the whole gamut. And we needed a way to operate with those modalities that is a little bit different from a traditional small molecule toolkit. So I've got here a chemical workbench for biopolymers. So what we have here is a little bit of a layout of what we're doing for small molecules, right? So compound design, synthesis, information cycle. We have a series of perceived gaps. Basically what this slide is telling you is that there are 5 steps that our discovery chemists take in hypothesis generation, design and selection, chemical synthesis, purification, registration, and then testing. And so this works really, really well for small molecules because we've got all the tools that we need. We don't have to worry about where the information comes from. We know where it comes from. We have all of the connectors that we have designed for this over the last few years. Problem becomes when we start to look at this from a biopolymer perspective, right? And so we need to fill some of these unmet gaps within these particular workflows, specifically for biopolymer chemists. So biopolymer chemists have different property calculations than small molecule chemists do. They have different synthetic procedures and synthetic tools. They have automated synthesizers that we need to integrate with. Their registration needs are different. Their purification needs are different. And specifically, their structure-activity relationship tools are very, very different. They always have to look at, if you take a biopolymer of 15 monomers, you basically have 15 small molecules that you're trying to optimize, as well as that 16th, which is the entire entity itself. So it's a very complex system for structure-activity relationship and very, very different from small molecules. The sub-goal, fill the unmet technology gaps as to fill the unmet gaps in the scientific workflow. So what were we looking for? We were looking for a COTS solution because we didn't want to deal with customization and all of the craziness that goes along with that. That doesn't exist. We're looking for a JavaScript solution. As the message of today, Java is really going away in all of the areas that I support. That didn't exist. And we're looking for a bulletproof perception engine. So as Roland was talking about, you have these structures that chemists are typically working with. Macrocycles, peptidomimetics, microproteins, a lot of them drawn out as fully blown out CTAB representation. So we need the ability to go from that to a sequence and then back to a structure as well. And it needs to be seamless for the end user. We don't want to disrupt our chemists' workflow by introducing a requirement that they register their biopolymers as a sequence, or they search for their biopolymers as a sequence, or anything along those lines. They're used to working with structures. It's familiar to them. They want to be able to do that. And of course, that didn't exist either. So we had some unmet gaps to fill. So the design, make, test paradigm for biopolymers. We're going to look at this from a scientific workflow first, and then we're going to talk a little bit about a quote-unquote technology stack that we're designing for this. We've got that little logo up there, the BioEddie logo for use or services or integration across the tools that we're working with. So our biopolymer design enumerator. So this is the first step that our chemistry modalities folks take. They look at modeled peptides or oligonucleotides, biopolymers of one kind or another, and design out a sequence that they want to investigate. Then they take a look at monomer locations around that biopolymer and say, I want to modify this particular location. And then they enumerate into a matrix and decide which ones to get synthesized. And this all get done manually right now in Excel. I literally have chemists who are copying and pasting within their Excel table 150 different sequences and modifying individual locations. It's not a very good way for them to spend their time. So we had to automate this and introduce some technology to make it a little bit easier for them. Second step is biopolymer-specific calculators. So leveraging that enumeration, that virtual library enumeration, and then applying specific calculators for the questions that they're asking, specific to biopolymers again, so they don't have the same tools that small molecule chemists have. They have specific calculators for their needs. We take the enumerated desired library and export the desired synthesis, the desired targets for synthesis. And we do this into a text format that's suitable for synthetic robots. Synthesize the desired library and register and purify successful syntheses. There's a little bit of a sub-step here that I'm going to talk about later on in the presentation, but the first part of it is thinking about biopolymer library synthesis and then taking individual biopolymers and performing modifications on them and doing traditional synthetic chemistry against larger biopolymers. So we'll talk about that in a moment. We then push these entities out to requested assays. So part of a drug program, you have requested assays for study. And then the assay results go into a Biopolymer SAR tool specific for my chemistry modality folks. And then central to this, this hypothesis is this Helm monomer library. This is what Roland was talking about, is that pharmas like myself, we have internal libraries that we don't necessarily want to share with others. We have ways of naming these particular monomers, and we have specific tools for distributing them for use across the organization. But all of these different steps within the workflow are pulling information from that Helm monomer library and going to be leveraging the services from BioEddie for use of that. Technology stack idea. So this is the Biopolymer Workbench is the first step. Second step is a new paradigm for technology stack within MRLIT. So we have many, many databases. Right? We've got databases for assay information, small molecules, our Helm monomer library, our biopolymers, everything that's associated with it in those multiple databases. Interrogating them is non-trivial, and so we're coming up with this idea and using the concept of a data platform, so our scientific information management data platform, to aggregate those databases. We're then going to expose services in this form for biopolymer property calculators on top of that platform, and then layer applications on top of that. And so we're going to have our biopolymer enumerator, synthesis request registration, assay data collection, Biopolymer SAR tools, as well as a plethora of other applications that go on top of that stack. So enumeration and design. So this is what I was talking about as our first step. So what I'm going to do is kind of walk through these steps and describe a little bit more about what we're doing with them. So you take your sequence, right, and this is going to be just a very simple linear sequence here, but the idea is extrapolated out to larger, more complex sequences as well. You have your biopolymer sequence, you have your variables, you have your monomers that you're going to be inserting into those locations within the sequence. You enumerate and get your virtual library. You then apply your biopolymer-specific calculations and predicted properties, and you get a synthesis selection. Do that synthesis selection. Robotic synthesizers, in this case mermaids, synthesize our biopolymers. We then do a mass-directed synthesis verification, and then purify and do a formal registration within our biopolymer database, and then apply those to assay requests and SAR analyses. So the enumeration tool is actually built already, and this is just a very quick and dirty POC of what we want to do so that our chemistry modality scientists can actually get into data collection and generation. So you launch your BioEddie from the top there. So you're going to generate your Helm input sequence using the BioEddie tools to do that. You choose your Helm file from your location. You decide what positions you want to change within that sequence, and you enter your monomer codes to be changed. You then populate out your matrix of enumerated biopolymer sequences. You can then export that out for more biopolymer calculators or direct to your synthesis requests or synthesis execution tools. We're going to introduce a third name to this puzzle here, and that's Discngine. And so our Discngine tools within Spotfire are leveraged heavily for small molecules. We want to do the same thing for biopolymers as well. So we want to take these and apply specific calculations and property calculators for biopolymers. One of them that we're going to be looking at is getting building block inventory as well, so not a calculator, but a tool that looks at our monomer inventory for the synthesis request to make sure it's present. And then something simple for multi-parameter optimization. You do your MPO calculations, and you're in Spotfire, so you go ahead and select your desired region of your scatter plot, and you get a list of Helm strings that are associated with it. Go ahead and populate one of those, grab one of those Helm strings, and you've got your sequence up on the top right, and then your structure below. Again, leveraging those services from BioEddie that Roland talked about. This is really key for the biopolymer scientists and chemistry modality scientists because, again, not all of them are used to operating in sequence mode, and not all of them are happy operating in structure mode. Depending on the size and complexity of those molecules, it's really going to vary from one scientist to another what the desired output is and workflow is for that scientist. We have to be able to support both. So design and enumeration, biopolymer virtual registration within BioReg. So we do this enumeration from the enumerator tool. We get our list of biopolymers that we're looking to register. We do a virtual registration. We're using our BioVia backend registration database for this. We then do some pipeline pilot scripting to pull out information from our Helm monomer library. This is the kind of tricky part. We have to translate this to another language that's specific for our synthesizers. And so we have to be able to understand the Helm monomer language as well as this Mermaid monomer language to push it out to our robotic synthesizers. We do that, and then we do our mass-directed synthesis verification, purification, assay request. So that's for libraries, biopolymer libraries. What about when you get you get a hit within your assay and you start to look at a specific scaffold and you want to do some structure-activity relationship studies on that specific scaffold? So we have to be able to do this on small scale for Singleton registration as well as SAR analysis. So this is an example of how a user wants to interact with the molecule that they're operating with. If you try to do this within a biopolymer registration system, you're going to have indexing problems because of the complexity of the structure, and so we're going to want to store these things as templates and sequences. So we do a registration and a perception of that structure and translate that into your Helm representation. And this is how the database is going to interpret and store that information. And it's all, again, seamless to the user. The user doesn't care about what happens. They simply want to be able to operate on that left-hand side as that structure. The second part of this is that there's an analysis done on the templates that are present. So there's no new templates perceived within this particular entity. And so the database registers it and stores it into the system. We want to introduce the idea of being able to identify new templates that are detected within the entity. So again, user interacts with it on the structure on the left, registers and does a perception, and new monomers are identified within the structure. And so we're asking the user to remediate that. We don't want to make the user go in and register new monomers within our Helm monomer library. We want to simply notify them that the Helm monomer library that we're using does not contain those monomers. Review which monomers are not present in the library, assign a 3-letter code per our business rules, verify with the user for accuracy, and push that to the central monomer library. And then again, complete that registration, and that's how your database interprets and stores that input. Totally seamless for the end user. A couple of clicks to register those monomers. The user doesn't have to worry about if they know what's in the monomer library or not. It's perceived, understood, and presented to the user as a new monomer. I've got a little slide malfunction there, that's all right. So biopolymer purification. So we do our mass-directed synthesis verification. We purify and do our formal biopolymer registration. And then our purification workflow is through an EKB lab notebook. And so that EKB purification workflow also has to interpret that Helm representation. And so that is also going to have to be connected to our Helm monomer library as well. Lastly, our SAR tools. So what we need to do here is our assay request and SAR analysis. And for Merck, that standard platform is Spotfire. And so we're going to have to integrate into our Spotfire platform for SAR analysis of biopolymers. So what's that going to look like? So I gave you a little bit of a mock-up of what we're looking like for design and for enumeration. This one is a little bit of a mock-up on what we're looking to get to for biopolymer-specific SAR tools. So again, disk engine panel on the left-hand side there for the tools that we want to expose. You get your selection of biopolymers into your data set. Perhaps you have some activity curves there. And again, you can go ahead and select that biopolymer and you get your sequence and your structure. Again, very important for all of our users to be comfortable within this particular setup so that they don't have to worry about manually switching between sequence and structure. We do have chemistry tools exposed, so if you want to do a structure search within your data set, you can do that much as you would for small molecules. Same idea for a sequence. Populated sequence search, and you can filter against your data set the same way you would for structure. Two potential biopolymer-specific SAR tools that we're looking at. One is what we're calling active monomers. And so we're going to identify within the sequence the chemical space that is either very good in one direction or very bad in one direction. So you've got your greens and your yellows and your reds to tell you where you want to look for structure-activity relationships. Matched molecular pair analyses is another SAR tool that is very popular with our scientists and does translate very well to biopolymers as well. So being able to identify which molecular pairs within a library are going to provide negative feedback from your assay results, so your red blocks there, your blue blocks for positive, and again, lining up directly to that sequence so you know which monomers within that sequence are appropriate, and then you can identify that from the sequence to the structure. All right, last little review again. So design and enumeration, biopolymer-specific calculations, enumerating our library for our synthesizers. We need to be able to export in a file format that is interpretable by our synthesizers. We need to synthesize our desired library, register and purify the successful syntheses, push those out to the assays, screen those libraries across the assays, get the results into our Spotfire visual, and again, all the while leveraging that Helm monomer library for interpretation and perception of those structures and sequences across the different tools that we have. A lot of folks at MRLT that helped with this, as well as our chemistry modalities group led by Rob Garbaccio and our modeling and informatics platform as well. Of course, ChemAxon for their terrific tools that are leading this space. EPAM is our developers, and Discngine has done some really good stuff for us as well. Thank you guys for your attention and for your presence here. And John, thank you for inviting me. I'm happy to take any questions.
So for the enumeration step, as I understand, you presented the case where there will be several variables in the sequence. In the mockup, I saw a single variation, and also it was specified by giving the index number of the position. Yeah. It made me wonder if it would be, I don't know if it exists or not, or if it would be interesting to allow in the editor to say that this is a variable.
It's next, yeah. Yep, it's a decent idea. I haven't heard that from my scientists yet. They were more interested in looking at cyclic connection points. So where do you make that macrocycle, and do you do it through a cysteine bridge or through some sort of chemical modification connection? But yeah, the ability to identify during the design is definitely a high priority for them.